if you've ever prepped for exams or, tried to survive a clinical rotation, you've seen it. That massive medical microbiology textbook,
Oh, yeah?
I think one author called it the Book of Pain, 1,000 pages of just dense, constantly changing facts.
That's a perfect description. And that feeling of just being buried in information is exactly what we want to fix today.
Right.
We're doing a deep dive into the absolute foundational knowledge, the high-yield stuff, pulled from sources like Patrick Murray's Basic Medical Microbiology. The whole mission is to give you a really solid, clear foundation.
And the key, which I love from the source material, is how we're organizing this. We're not doing it by disease.
Right.
We're organizing it by the organism itself, bacteria, viruses, fungi. Because if you really understand the microbe, you can build a differential diagnosis so much faster. It gives you the mental framework.
That's it, exactly. So let's start with that framework. The 4 big kingdoms of microbes that matter in medicine. You can think of it as a scale, really, of increasing complexity.
Okay, so let's walk up that scale. At the simplest end, we've got viruses.
Viruses. Then a step up, you get bacteria.
Okay.
Bit more complicated after that are the fungi.
And then at the top, the most complex things we deal with are the parasites.
That's the hierarchy. And viruses are just masters of simplicity. I mean, they're nothing more than some nucleic acid, some proteins, and maybe a lipid envelope.
And they can't do anything on their own?
Nothing. They're completely 100% dependent on a host cell to replicate, to survive. They're basically just genetic hijackers.
So for a quick ID in the lab, what's the one thing that defines a virus?
It's genome. It's simple. A virus has either DNA or RNA.
But not both.
Absolutely never both. And that one fact dictates almost everything about how it behaves.
Okay, so if viruses are that simple, moving up the ladder to bacteria, that's a whole new world. We need a different toolkit.
A completely different toolkit. Bacteria are prokaryotes, unicellular, but they have no internal membrane-bound structures, so no nucleus, no mitochondria.
None of that complex machinery.
Right. And their defining feature is the cell wall that surrounds them.
Which brings us directly to, the most fundamental tool in the microlab, the Gram stain.
The Gram stain, yep.
Yeah.
It's a brilliant, simple test that splits almost all bacteria into 2 giant groups based entirely on that cell wall.
So Gram positives, they have that really thick wall.
A thick peptidoglycan layer. Yeah, it just soaks up and holds onto that first crystal violet dye.
While the Gram negatives, they're trickier.
Much trickier targets. They have a thin peptidoglycan layer, but it's covered by this outer membrane. It blocks the stain. And it's also an extra layer of defense against antibiotics. After that, you're looking at shape spheres, rods, spirals, and how they're arranged.
That basic map is so critical. All right, let's jump into some of the big players, starting with those aerobic gram-positive coccus. We're talking Staphylococcus, Streptococcus, and Enterococcus.
These are everywhere, and their clinical importance really comes down to either their toxins or their resistance.
So take Staphylococcus aureus. What makes it so bad?
Well, it's incredibly versatile. It can cause a simple abscess, sure, but it can also cause these devastating systemic diseases. And it does that by releasing superantigens, like the one that causes toxic shock syndrome.
And the resistance part of the story is pretty grim.
It is. The CDC figures in the U.S. alone, you're looking at 80,000 MRSA, that's methicillin-resistant Staph aureus infections a year.
Wow.
And over 11,000 deaths from that. That tells you right away you have to be thinking about resistance with SREs.
Okay, now what about Streptococcus pneumonia? Is that a resistance story too?
It's more of an evasion story. That's a good way to put it. S. pneumonia is the #1 cause of community-acquired pneumonia. We're talking 400,000 hospitalizations a year.
And all those ear infections in kids.
7 million cases a year. Yeah. Its secret weapon is its polysaccharide capsule. It's like a shield that lets it hide from our immune cells. The problem is there are nearly 100 different types of that shield, 100 serotypes, which makes vaccination a real challenge.
So if strep complicates things with serotypes, Enterococcus just complicates things with brute force resistance.
It's the poster child for resistance in the gram-positive world, yeah. I mean, vancomycin resistance used to be rare. Now, for Enterococcus faecium, over 90% of them are VRE, vancomycin-resistant enterococcus.
And your treatment options just plummet.
They shrink to almost nothing. It's a huge problem.
Okay, let's switch gears to the gram-positive rods. And we can split them into spore formers and non-spore formers. Let's start with the famous one, Bacillus anthracis.
Anthrax. It's a zoonotic disease, fundamentally.
Hmm.
Humans are just accidental victims, usually from contact with animal products or, and this is the scary part, aerosolized spores.
The bioweapon potential.
Exactly. The spores can survive for decades, and the disease progresses so quickly.
How does it cause that kind of rapid collapse you see with inhalation anthrax?
It's a cocktail of virulence factors. It has a capsule to protect it, and then it has these three toxins: protective antigen, edema factor, and lethal factor. The combination just overwhelms the system. You see this hemorrhagic mediastinitis, bleeding in the chest, that's just catastrophic.
And in total contrast, you have a non-spore former like Listeria monocytogenes, which is more of a public health threat from our refrigerators.
Right. Listeria is all about tolerance. It can hide inside our own cells, and it grows happily in the cold and high-salt environments.
So deli meats, soft cheeses.
That's why you see these multi-state outbreaks linked to processed foods. And it's a particularly high risk for pregnant women and anyone who is immunocompromised.
Okay, one last group within the rods. The acid-fast bacteria, Mycobacterium. What does acid-fast actually mean?
It just means they resist being decolorized by acid during the staining process, and that's all because of this waxy substance in their cell wall called mycolic acid. The two big ones to know are Mycobacterium and Nocardia.
And Mycobacterium tuberculosis is still a massive global problem.
A huge problem, 9.6 million new cases every year. But this area also gives us a huge public health success story with M. leprae, the cause of leprosy.
Right.
It's been eliminated as a public health threat from 119 countries since 1985, mostly just by getting free, effective drug combinations to people. It shows you what's possible.
Okay, let's make the jump to the gram-negative rods, starting with that enormous family, the
The
Enterobacteriaceae.
gut bacteria. Yeah, they're defined by what they can ferment. They grow fast, they all ferment glucose, and they're oxidase negative.
And for a clinician, something like lactose fermentation is a really quick diagnostic clue, right?
It's a great triage tool. If you get a stool sample from someone with diarrhea, you want to know if you're dealing with a pathogen. Things like Salmonella and Shigella don't ferment lactose, so you can use a special agar plate that changes color to spot them right away.
And we've seen how just one nasty strain can cause huge problems, the E. coli outbreaks.
Absolutely. That 2006 outbreak of Stec 0157 from spinach is a perfect case study. A tiny amount of contamination led to widespread illness and a really high complication rate. 16% of people developed hemolytic uremic syndrome, HUS, which is a severe kidney failure.
And still in the gram-negatives, you have organisms like Klebsiella pneumonia, which are just notorious for developing resistance.
Klebsiella has this big, goopy mucoid capsule that makes it really hard for the immune system to grab onto. But the real threat now is its ability to produce carbapenemases, KPC.
These are enzymes that just chew up our best antibiotics.
They destroy almost all our beta-lactam antibiotics. It can make these infections virtually untreatable.
So beyond the fermenters, we have the aerobic non-fermentative rods, the opportunists.
The ones that thrive in hospitals, their main asset is just resilience. They can survive on almost nothing contaminated, disinfectants, hospital sinks, you name it. And they're naturally resistant to a lot of antibiotics.
And the scariest one in that group has to be Acinetobacter baumannii.
It's a terrifying example of how global events can change microbiology. Multi-drug-resistant Acinetobacter really exploded and spread globally with the military conflicts in Iraq and Afghanistan.
And what's the most common one we see in hospitalized patients?
That's going to be Pseudomonas aeruginosa. It has a whole arsenal of toxins, but its ability to cause disease really depends on the patient. In a healthy person, it's usually harmless.
But in someone on a ventilator or with burn wounds?
Its virulence just gets fully unleashed. It's all about opportunity.
All right, last group of bacteria. The spiral-shaped ones. These can be tough to diagnose.
They really can be. They're often too small to see easily. They have special growth needs, so we often have to rely on serology, looking for antibodies in the blood instead of culture.
And a key one here is Campylobacter jejuni.
Most common cause of bacterial gastroenteritis in the world.
Uh.
But the real clinical alert with Campylobacter is the potential complication. It can trigger Guillain-Barré syndrome, that post-infectious paralysis.
And then there's the one that causes ulcers.
Helicobacter pylori. The discovery that this bacterium was linked to gastritis, peptic ulcers, and even some stomach cancers. That completely changed gastroenterology.
Okay, so we've covered the prokaryotes. Now we're shifting gears into a whole different level of complexity with the eukaryotes, starting with fungi.
Exactly, and this is a big leap. Fungi are eukaryotes, so they have a real nucleus, organelles. just like our own cells.
Which makes them hard to treat, right? It's hard to find a drug that kills them without hurting us.
That's the challenge. But they do have some unique features we can target. Their cell wall is made of chitin and glucan, not peptidoglycan. And the big one, their cell membrane uses a molecule called ergosterol instead of cholesterol.
And that's the target.
That's the bullseye for most of our best antifungal drugs. We can attack their ergosterol, disrupt their membranes, without touching our own cholesterol-based ones.
So looking at the opportunistic fungi, who are the usual suspects in the clinic?
Number one is Candida albicans. It causes everything from minor thrush to deadly bloodstream infections or fungemia. The emerging problem, though, is resistance to our main drugs, the azoles, especially in species like C. glabrata and C. cruce.
And what about fungal meningitis?
That's usually Cryptococcus neoformans. It's a yeast that makes this huge protective capsule. You get it from inhaling spores, often from places with pigeon droppings, and it almost always causes meningitis in people who are immunocompromised.
All right, finally, we've reached the top of the complexity ladder. Parasites. We'll start with the single-celled ones, the protozoa.
And globally, there's one that stands above all others. That's Plasmodium, the parasite that causes malaria.
The numbers are still staggering.
200 million cases a year. It just highlights why chemoprophylaxis, taking preventive medicine, is absolutely non-negotiable if you're traveling to an area where it's endemic.
And here at home, what's the most common cause of big waterborne outbreaks?
That would be Cryptosporidium. It causes terrible diarrhea, and it spreads so easily. We see huge outbreaks from contaminated swimming pools or drinking water, but also from things like sick food handlers. That's a tough one to control.
And our last group, the helminths, the worms. Multicellular, complex life cycles. Some of them get huge.
Exactly. I mean, the intestinal roundworms, like Ascaris, probably infect about 1 billion people. But the one with the biggest clinical alert, the one you have to be vigilant for, is a tiny roundworm called Strongyloides stercoralis.
What's the specific danger with strongyloids?
So this parasite is common in tropical areas, and it can cause a latent, low-level infection for years. The source material is very clear. You must screen for it before you start someone on immunosuppressive therapy, especially corticosteroids.
Why? What happens?
If you suppress the immune system, the parasite goes into overdrive. It's called hyperinfection syndrome. The larvae replicate uncontrollably, spread all through the body, and it often leads to a fatal septic shock.
That is a terrifying interaction between a host, a pathogen, and a treatment. What about the cystodes, the tapeworms?
Cystodes are the flat, ribbon-like ones, and they can range from a few centimeters to over 10 meters long.
Wow.
The most dangerous is Taenia solium, the pork tapeworm. If you eat the eggs, not the worm in the meat, but the eggs, the larvae can end up in your tissues. If they get into the brain, it causes neurocysticercosis, which is a major cause of seizures worldwide.
This whole discussion just proves the value of that initial method. If you know the organism's basic classification, is it gram-positive? Does it have ergosterol? Is it a parasite with this kind of life cycle? It just immediately helps you narrow down what's going on with the patient.
It sharpens your thinking instantly.
Yeah.
And we have to end by saying, this field never, ever stands still. We have emerging viruses like SARS and MERS. We have constant evolution of drug resistance. We have diseases like pertussis coming back because adult immunity is waning. It requires constant learning.
And to really lock that in, let's finish with the clinical challenge that pulls these principles together.
Okay, imagine this. A 46-year-old Cambodian immigrant comes in with diffuse muscle aches. He started on corticosteroids for what they think is necrotizing myopathy. A month later, he's in septic shock with E. coli in his blood, he has hemorrhage in his lungs, and they find numerous larvae in his breathing tube secretions. Based on what we've just talked about, why was starting that immunosuppressive therapy so dangerous for this patient? And which parasite was responsible for this absolute catastrophe?
That's a perfect taste to think through, connecting the dots between latent infection and immune suppression. It really shows why this foundation matters. That's a wrap on our deep dive into basic medical microbiology. We hope you feel a little less overwhelmed and a lot more equipped to understand this world.